During the past decade, we had enormous improvements in understanding the genetic basis of acute lymphoblastic leukemia, with currently 25 different subtypes characterized by distinct genetic alterations with a distinct clinical outcome. However, all these studies were done by bulk sequencing that, although very powerful in detecting these drivers, has a limit since it cannot detect intrasample tumor heterogeneity and cannot infer cell of origin, which may be important to prevent or understand better therapeutic responses. Thus, here we used the single-cell RNA-seq, which has the power to dissect intratumor heterogeneity and to precisely measure cell developmental states within B-cell ALL samples. We know that B-ALL may be a rise from progenitors already committed toward B-lymphoid lineage. However, there are also evidences that the origin may be immature and so involving more stem cells and early progenitors, or even later during B-cell development at the very late stages. However, understanding how the different cell states correlate with the genetic drivers with the clinical outcome, this has been poorly understood. Here, through the development of a human reference map of B-cell development, we were able to clearly characterize different cell states along these trajectories and use these annotations to refine cell type enrichment in different VLL subtypes. These enabled us to identify subgroups that are enriched with early lymphoid progenitors and even some stem and more immature populations, subtypes of VLL that are instead enriched with late pre-B cell abundance, and most of these subtypes they have a consistent abundance of pro-B cells. So what this does mean means that currently we are using, for example, immunophenotype for determining the lineage of a certain leukemia subtypes. Here we not only have information on the expression of several surface markers, but we can really detect rare populations that may be meaningful in terms, for example, for disease progression of responses to therapies. Indeed, we were able to show that, for example, in certain subtypes such as BCR-ABL or KMT2 area arranged and ZNF384 rearranged BLL, we couldn't detect a subgroup with a very early lymphoid signature expression that is similar to that we found in mixed phenotype leukemia. So two entities that are diagnostically different, but they have the same cell composition and the same gene expression profiles. And so this means that analysis by single cells and especially by cell developmental state can help in refining the specific subtypes and can help in predicted outcome. Indeed, we were able to show that those subtypes with an enrichment of these early populations that have a worse outcome compared to the others. And so I'm sure we will hear more in the futures when many additional samples will be analyzed. We can have a better understanding of the transcriptional signature, cell developmental states within each leukemia subtype and tailor better therapies and eventually predict relapse.